So this will now be on tape, but this is the lab. This is what we're going to do. So we bought Piglo from a company called BioRed. Then through a process we called transformation, where we put the plasmid into a bacteria overnight, where we grew up those bacteria, and mini prep, where we took the plasmid back out, we should end up with the plasmid Piglo. And we should have lots of it. All right, so we are going to look at your samples on Friday. You're going to nano-drop your Piglo. Did you already nano-drop your Piglos? Yes, you've nano-dropped your Piglos, and we've looked at 260, 280s. You have images of the graph so we can tell how clean it is?
I don't know if they're saved.
We recorded the numbers, but we didn't take any of the graphs down.
Okay. Always take a picture of your data. Just take pictures of stuff. A lot. All right? Okay, so what we're then going to do is, through a process called DNA replication, we're going to build a small, we're going to build a small piece of DNA. And that's called a primer. Now the primer is going to attach to Piglo because it will be the same sequence. So don't worry about this too much yet. But what we're going to do is make a mistake in the primer. So the primer is not going to actually stick to Piglo perfectly, but it'll have a little bump there where the mistake is. We're going to build another primer in a similar way, having the same mistake. And then what we'll do is we'll build and we'll copy the new the Piglo off of those primers. So, imagine this. I've got a book. It's let's just call it the alphabet. It's a page of an alphabet, right? Okay, so I put down 3 letters that should be B, C, D, corresponding to the letters B, C, D, except I write B, A, D. Okay? So then I copy the rest of the text. Now I have a piece of paper that has B, A, D and the entire alphabet in it. So basically an A instead of a C at that one position. So let me show you exactly what we're going to do here. Awesome. Okay, so let's say my plasmid is really long and it's linearized. So let's say this part is T, C, 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 G, A, C, G, and this is A, G, C, T, G, C. What I'm basically going to do is build a primer that is A, G, C, C, G, C. Do you see the mismatch here? A normally binds to what letter?
T.
T. Okay. And what's here?
C.
C.
Yeah.
So it won't match up perfectly. I'm going to build a primer in the other direction, this time with a G there. Oh. Yes?
Wouldn't the 2 primers preferentially stick to each other instead of the DNA?
Very good question, and yes, they will. But we're going to manipulate it in stoichiometrically so that some will at least get here, and then we'll get rid of the small pieces. That's a very good question. So, once we have our primers down, we're going to actually build the rest of the DNA, just copying. And don't forget this is a loop, so it'll come back on itself. And this is a loop, so it'll come back and copy on itself. So now we're going to have these old DNA in black and new DNA in green hybrids. We're going to digest using an enzyme, a nuclease. What does a nuclease digest, do you think? Nucleic acid, right? DPN1 digestion. Okay, so it turns out that this enzyme, DPN1, can digest naturally made plasmids. This original plasmid, bacteria made for us. The green plasmid, we made in the lab. Yes?
How does it distinguish?
So when bacteria makes plasmids, it actually adds small methyl groups to the outside of the DNA. DPN1's active site has that space for methyl groups. So non-methylated DNA will not be destroyed. In the end, we'll have pGlo like that, except it will have a single mutation change. In this case, we've gone from an AT to a CG.
So we're basically like making an evil twin and then like getting rid of the original.
Mm-hmm. And then, through the process of transformation, we're going to put this back into bacteria. No, this is I need a different shade for bacteria.
There's some reds down here.
Wait, Luke.
Yes?
So how did you get from the bump to no bump?
So I destroyed the original bacteria. The original plasmid.
Wait, with the digestion thing?
The digestion will destroy the original plasmid. Yes. Nico.
So the bump is the color difference, pretty much.
The bump will be essentially the
So
color difference.
that bump is going to be, so our primer is going to be part of the GFP, right?
Primer is going to be part of the new.
But the bump
It won't
is
be
gonna
GFP. It'll be YFP at this point.
The YFP is what it's called when it's new.
Yes.
So our bump is,
Yellow.
but GFP is more than 7 nucleotides, right?
Correct. So GFP is this like a chunk of this whole thing.
And our primer is just the part of GFP that's the color.
Correct. Just the part that is going to change the color.
Okay, so my question is, who, how, and why do they figure out? No, sorry, just who and how. No, just how. How did the scientist or person who figured out what part of GFP indicates the color?
Well, he won the Nobel Prize for it, and it is in your canvas in a section labeled something like.
You're at 1%.
Oh, thanks.